Hello everyone, this is Dr. Maha Abdurrahman. I'll be solving today one of the most challenging questions about diabetes, analyzing curves, using paper 2 from the Oxford AQA exam, November 2019. And I'm gonna head to question 5. Question 5 is touching base for the skill of analyzing graph, which is required for all the exams in IGCSE level and in A level as well. Now, the really secret behind how to do a good analysis for graph is reading the question, okay? You need to really spend a good time reading the question, understanding what exactly are your parameters on the x-axis, on the y-axis, what exactly do we need to measure. So let's check question 5. Scientists investigated the relationship between concentration of insulin and glucose in the blood of healthy people, and that was the method. So they took a blood sample from 50 volunteers, so those are the number of volunteers used. Each volunteer drinks a solution containing 50 grams of glucose, and then the blood sample was taken every 30 minutes for the period of 2 hours. Measure the concentration of insulin and glucose. So insulin and glucose concentration was measured in each sample. Then they calculated the mean values for blood insulin concentration and for blood glucose concentration for the volunteers at each time intervals, which was 30 minutes. So it's quite self-explanatory, the method, very clear, and figure 6 is showing you the results of the graph. You can see here on the x-axis the times in minute, and it's in 30 minutes interval for 2 hours, as you see. On the right you will have the mean glucose concentration in millimole per decimeter cube, okay? That's on the right. On the left you will see the mean blood insulin concentration in millimole per decimeter cube, and clearly the solid line is for the glucose, the dotted line is for the insulin. It's really important to know which is which is also self-explanatory here on the graph. Now let's check the first question. It needs you to calculate the rate of increase in the mean blood insulin concentration from 0 to 30 minutes. So I need to check here on the x-axis from 0 to 30 minutes, okay? And then they need the rate of increase in the mean blood insulin concentration, which was clearly moving from 0.2 at the time of 0 minutes up till, if you move, it's gonna be, if you do your division, it's gonna be 0.086. That's the concentration at 30 minutes. So you just need to go up, okay, and then straight line, it will give you the reading, okay, 0.086. Since every square here is 0.02, as you see, right, as they are 5 squares, so that's a 0.02, 0.082, 0.084, 0.086. Every square is 0.002. Okay, now with that being said, I would write for the rate of increase, the rate is always the change in the y-axis over the change in on the x-axis. So that's gonna be on the y-axis 0.086, as I mentioned, minus 0.02 divided by the difference on the x-axis is obviously the 30 minutes. Guys, to calculate the rate of anything, you need to have the change in the y-axis over the change on the x-axis, and then if you do the math, it's gonna be 0.002 millimole per decimeter cube. That's 2 marks. 1 mark for showing your work and 1 mark for the final answer. You don't need to write the unit because it's obviously written here. Now let's move to the second question, talking about describe the changes in the mean blood glucose concentration during the investigation. Whenever you see the word describe, please don't explain. Don't try to show them that you know why the trend is moving up, is moving down, increasing or decreasing. You just need to describe. That's it. Okay? So if I check here, the mean blood glucose concentration change during this investigation, and I need only to be focusing on the glucose. So if you see here, it's increasing from 0 to 30 minutes, right? Then it's decreasing from 30 minutes to 90 minutes, and then it's constant from 90 minutes up to 120 minutes, okay? And that's all what we need to write. That's it, right? So basically, as you see, till here, I just need some skills to answer this question. I didn't use any information up till now. So describe the change in the mean blood glucose concentration during the investigation. I would say from 0 to 30 minutes, there was an increase, okay? Increase in the glucose level. That's the 1 mark. From 30 minutes to 90 minutes, there's a decrease in glucose. Again, you don't need to mention the reason. And then, that's the 2nd mark. Then finally, from 90 to 120 minutes, there's a plateau, right? What I mean by plateau means no increase, no decrease. It's a constant. It's constant. Okay? As simple as. Then if you check the next question here, you guys gonna explain. Explain the changes in the mean blood glucose concentration. Here it's your time to show your knowledge. So basically we're using your skills. In the first 2 questions, here it's time to test our knowledge. Why this trend is the way it is. And it's 3 marks, so basically you're gonna explain every trend that you see here. So basically why there was an increase in the glucose at the very beginning from 0 to 30 minutes, because as you ingest the 50-gram glucose drink, right? As you ingest the glucose drink at the very beginning, okay, it's being absorbed from the intestine, right, into bloodstream. This is why there is an initial rising, okay? Then, after that, there's a decrease in the glucose, right? What's the reason of this decrease? So that's the 1st mark. The 2nd mark, you should account for the decrease in the glucose level. And that was because insulin was released. If you check back here, guys, the curve, as you see here, after the glucose has been increasing, insulin has been released, and so consequently glucose is being decreased because insulin, I'm gonna write this here, insulin is what convert glucose into glycogen. Why does this happen? Because I need to lower the amount of glucose in the blood. So glucose in the blood, when insulin is produced, is going to be stored as glycogen in liver and muscles, because basically I need to get this level of this amount of glucose down, and that's why technically the glucose is decreasing. I hope that was clear. Then definitely the last part, why glucose stayed back to normal, because it had been already decreased to the amount or the normal amount, and that's why you see here there's a decrease in the amount of insulin, because we don't need high insulin anymore, because already the glucose is its normal level. Okay. Also, something else that also I want to make clear, insulin does not only push glucose or convert or store glucose into glycogen in the liver muscle, insulin also promote or encourage cells to absorb glucose. So basically insulin, it push the glucose inside the cell, so it makes the cells absorb more glucose. Why does this happen? Why does it push or allow cells to absorb more glucose? Because basically cells will need to utilize this glucose in aerobic respiration to get energy. Okay, now let me now go back to what I'm gonna write here. So I would write exactly what I explained. I'm gonna write it in here, that because insulin is released, definitely insulin is released from the pancreas into the blood. Okay, so this referring to insulin is the second mark, and then referring how glucose will be decreased. So this will move glucose inside the cells, or you can also say that it will convert glucose into glycogen. In both cases, this will lower the glucose in blood back to normal level. I hope that was clear, explaining of the insulin role. Now, still on the same question, the volunteer must remain seated throughout the test. That's in interpreting information. Why the volunteer must remain seated throughout the test? Why this is important? Because if this volunteer is moving, obviously he will need to use more and more glucose in aerobic respiration to provide him with energy, which will make this test not really valid, right? Because I need to measure the amount of glucose in the blood in a fair way. So if this person is moving, the experiment is not valid. Why is it not valid? Simply because activity would increase the glucose uptake by cell, right? Uptake by cells, because they basically need that in the aerobic respiration, okay? Reducing the glucose level in the blood, okay? Now, question 5.5. This is kind of a hard question. It's completely indirect one. The blood sample were taken from capillaries. Explain how blood glucose concentration is lower in veins than in capillaries. Explain why the blood glucose concentration is lower in veins than in the capillaries. And if you have the information that capillaries, let me draw a simple drawing here to explain this. Now I have, whenever you have body organs or cells, you will have capillaries, right? Those capillaries are basically coming from arteries. Those are the capillaries, right? And then blood it's gonna come next to the organs and cells. Those capillaries will supply those organs and cells with oxygen and glucose. And when these cells do aerobic respiration, they will provide the same capillaries again with carbon dioxide. These capillaries will join to form veins. Okay? Those veins would be full of carbon dioxide waste, right? That means less glucose, because basically glucose will be used by those organs and cells in the aerobic respiration. That's a very quick explanation for it. So it's a 3 marks question, no doubt, because there's a lot of explanation. That was a very like a summary for how to answer this question. So I would say that blood from capillaries will move into vein, right? Will move into veins, and then from already, that's the first mark. Now already cells and organs have used the glucose for aerobic respiration, right? For aerobic respiration. So that's why the blood returning back to vein would have less glucose. Now, last question, 5.6. Type 1 diabetes is a condition which leads to high blood glucose concentration. Part of the treatment of type 1 diabetes is carefully controlling the diet. Suggest why a person with type 1 diabetes should eat food containing starch rather than glucose. And if you have the information that starch is hardly digested to give glucose, while glucose is already in the glucose form, right? So it will higher the blood sugar in a faster manner than if these people are eating starch. And this is really the idea of this question. So I'm gonna write it down. Very simple, very easy. So starch would take more time to be broken, than glucose. Oh, it's gonna be broken down into glucose anyway, right? But it's gonna take more time, right? So they would be less or slow increase in blood sugar or in blood glucose level than if they eat glucose directly. This will bring me to the end of this tutorial. I hope it was simple and easy, explaining the question about insulin, glucose, how to tackle analyzing graph questions. And obviously, as you see, you need to acquire not only information, some skills that's gonna help you to answer the IGCSE, particularly Oxford AQA. They really stress on the skills, how to improve those skills, how to acquire those skills at the very beginning, as important as acquiring information. So it's not only information, you need as well to build and work on your skills. Thank you so much. I hope this was a beneficial video. See you next time.